Welcome to the Dr. Hüfner laboratory in Hergatz in the Allgäu. With our many years of expertise in the field of milk and cheese, we support direct marketers and companies with challenges and new developments. Every day starts with the receipt of cheese samples and the verification of the sample accompanying documents. After labeling and temperature measurement, we create an order with a photo in the system. Then, the samples are temporarily stored in the lab refrigerator. Here, the samples undergo a visual inspection and are weighed. During this process, the investigation parameters are determined and a preparation for further examination is made. Depending on the case, our microscopy department examines the samples immediately and plate growth is established, especially for samples with visible mold. After the initial examination, the samples are taken by an elevator to our PCR laboratory. Here, the samples are scanned into the system and pipetted accordingly for the PCR run. After 1 hour and 20 minutes, the results of the PCR analysis are available. For the chemical analysis, the cheese samples are first crushed using a mixer or ultraturax. Next, the sample is weighed for the various analyses that are carried out according to the customer order: dry matter, fat content, protein content, salt content, and sugar determination like lactose or galactose. For dry matter determination, the sample is weighed on a special foil using an analytical balance. The sample is evenly distributed with a foil roller. Then the foil is opened like a book and the edges are bent upwards. This preparation allows for drying in an oven at 102 degrees Celsius for 2 to 3 hours. After weighing the dried foil, the dry matter content is calculated. The fat content is determined using a butyrimetric analysis. After the sample is broken down with sulfuric acid and centrifuged, phase separation is achieved by adding amyl alcohol. The fat content is then directly read as a percentage on the scale of the butyrometer. To determine the protein content, the sample is digested with sulfuric acid and distilled using a Büchi device. Cooking with caustic soda releases ammonia, which is captured by boric acid, causing a color change from purple to green. Then, the nitrogen content is calculated by titration with hydrochloric acid. The salt content is determined either with a hydrometer as a quick method or more accurately by the Erbacher method. In this process, chloride ions are precipitated with silver nitrate. After digesting the proteins and titration with ammonium thiocyanate, the exact calculation of the salt content is carried out. For the enzymatic determination of lactose and galactose, the sample is first deproteinized, the pH value is adjusted, and the solution is filtered. The test solution is pipetted into cuvettes with a test kit and measured by the photometer. The calculation of the sugar content is based on the extinction values in a Microsoft Excel spreadsheet. The measurement of water activity is done by placing the sample in a special measuring device. The values are automatically determined and documented by a software program. The microbiological analysis includes the examination of the cultured plates under a microscope and the differentiation of germs using MALDI-TOF. A highly advanced analytical laser is used here for the mass spectrometry of chemical compounds. To avoid contamination, the cheese samples are evaluated by our sensory department using the DLG 5-point scheme only after all examinations are complete. Appearance, consistency, smell, and taste are evaluated, and points are given on a scale from 1 to 5. Finally, the test reports are personally inspected and signed by Dr. Hüfner. This is how we guarantee for the quality and reliability of our work. For over 30 years, the Dr. Hüfner laboratory has been producing germ count and cell count standards for the dairy industry. The quality of the standards is continuously monitored and tested using Voss Baxomatic with reference samples. Later, specially developed extra-high goat cell count standards were added to the portfolio. Our standards are available with 3 measurement levels and allow flexible and precise quality control of measuring devices. Every 4 weeks, we ship our batches to all of Europe and South America. To ensure the samples remain optimally cooled during transport, we include ice packs. This ensures an unbroken cold chain, even to our customers in Brazil, the Azores, or the Canary Islands. Our customers benefit not only from detailed batch analysis, but also from our decades of expertise, a reliable basis for the objective assessment of the measurement accuracy of your devices. Last but not least, our service offering also includes DN hygiene testing for commercial dishwashers. This involves checking whether the dishwashers reliably remove germs and residues. For this purpose, glasses are first cleaned and then treated with a special test solution. The resulting liquid is prepared for laboratory analysis to check the hygienic effectiveness of the washing process. Laboratory Dr. Hüfner, your Dairy Science Institute.